How does the brain interpret light entering into the eyes? The brain interpretation of light enters the eyes is a complex process involving the visual system, which transforms light energy into neural signals that are processed to form visual perception. This process integrates optics, neurobiology, and cognitive processing, building on your interest in IBNL Haytham's work on vision and Vermeer's use of light. Below is a concise explanation of how the brain interprets light, tailoring to provide a clear overview with connections to your previous question. Overview of the process. Light enters the eyes, is focused onto the retina, converted into electrical signals by photoreceptors, and processed through a series of neural pathways in the brain to produce the perception of shapes, colors, motion, and depth. IBN Al-Haytham's book of optics was the first to correctly describe light traveling from objects to the eyes in intromission theory, laying the groundwork for understanding this process. Step-by-step process. Number one, light enters the eye. Light from an object, Vermeer's luminous girl with a pearl earring, passes through the cornea, which bends, refracts it. Then through the pupil controlled by the iris to regulate light intensity, the lens further focuses light onto the retina, a thin layer at the back of the eye. IBN Al-Hatham's experiment with lenses and pinhole cameras helped explain how light forms an inverted image onto the retina, a principle still central to optics. Number two, photoreceptors convert light to signals. Beep, 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 beep. The retina contains photoreceptors rods, about 120 million sensitive to low light, responsible for night vision, and cones about 6 million sensitive to color and detail, concentrated in the fovea. Cones come in three types, detecting red, green, or blue wavelengths, enabling color vision. When light hits photoreceptors, pigments in rods, photopsins in cones absorb photons, triggering a chemical reaction that converts light energy into electrical signals. Bing! Phototransduction. This process is sensitive to wavelengths, like the blue and red light Vermeer's painting reflects. Chlorophyll, from your earlier query, absorbs similar wavelengths, red and blue, reflecting green, chlorophyll, which is why plants appear vibrant to our cone-driven color vision. Number three, signal processing in retina. Photoreceptors synapse with bipolar cells, which connect to ganglion cells, whose axons form the optic nerve. Horizontal and endocrine cells modulate signals, enhancing contrast and edge detection. The retina performs initial processing, such as detecting light gradients, Vermeer self-shadows, and motion, reducing the data sent to the brain. This is why L. Haytham emphasized the retina's role in vision beyond mere light reception. 4. Transmission to the brain. Ganglion cell signals exit to the eye via the optic nerve, carrying electrical impulses to the brain, the optic disc, where the optic nerve exits, lack photoreceptors, creating a natural blind spot. Signals reach the lateral geniculate nucleus, LGN, in the thalamus, which relays them to the primary visual cortex, V1, in the occipital lobe. The LGN organizes visual input, separating left and right visuals. Light from the left side of the painting goes to the light from the left side of the painting goes to the right side of the brain. Number 5, processing the visual cortex. Primal cortex V1, located in the occipital lobe V1, maps the visual field, detecting basic features like edges, orientations, colors. For example, it identifies contours in Vermeer's Pearl of the Window's light gradient. Higher visual areas V2, V4, V5, and T. These regions process complex features. V2 refines edge and texture perception. V4 specializes in color, crucial for perceiving ultramarine blues, like in Vermeer's painting. B5 detects motion, less relevant to static painting but key for films. 6. Integration and perception. The brain integrates visual data with memory, attention, and context in areas like the prefrontal cortex and temporal lobe, forming a coherent perception. For example, recognizing girl with a pearl earring, as a portrait involves matching visual input to stored knowledge of faces and art. Hib al-Haytham insight into perception as psychological process combining sensory input with mental interpretation anticipated this step as he noted the brain judges visual data. Emotional response, like awe at Vermeer's glowing light, involve the amygdala and the limbic areas trying vision to feelings. Key water and influences. Binocular vision, the brain combines slightly different images from each eye, perceived depth stereopsis, enhancing the three-dimensional feel of Vermeer's scenes like the receding walls in the music lesson. Color perception, cone sensitivity to red, green, and blue wavelengths allows the brain to interpret Vermeer's vibrant palette. With V4 processing the interplay of light and color, adaptation and context. The brain adjusts light conditions, example, bright versus dim galleries, via pupil dilation and neural adaptation, ensuring consistent perception. It also uses context, like recognizing a painting's historical significance to shape interpretation. Heart-mind coherence connection. The serene light in Vermeer's paintings may trigger parasympathetic responses, promoting calm and focus during viewing. Challenges and variations. Visual disorders, conditions like color blindness, cone deficiencies, or macular degeneration impair light interpretation, altering how one might perceive Vermeer's color details. Illusions. The brain can misinterpret light as in optical illusions, where context tricks perception, a phenomena studied in Al Haytham studied in his work on visual errors. Significance. The process enables humans to experience the world visually, from practical tasks to aesthetic appreciation of art, like Vermeer's, where light subtle play pointilleux, soft gradients, captivates the brain's visual circuits. IBNL Haytham's experiment with light paths through the eye directly informed modern understanding, bridging his 11th century insights to neuroscience. The process also ties to quantum physics from Planck, as light particle nature photons initiates phototransduction, a quantum event. Okay, beautiful, beautiful. Thank you, girl. This is very interesting. We learned a lot.